Good morning, students. Good morning to all. Today, ninth day of the section. Students, we are having Facebook Live and YouTube Live also. Please check it there. Anyone facing problem in YouTube near meet means you can join there also. Within 2 minutes, sir will join students. Can you? My voice is audible or not? Please respond in the chat box, students. If my voice is audible, within 2 minutes, our section will be start. Please check YouTube Live and Facebook Live is also going on. Please check in the students. Once again, I am announcing today is ninth day of the section. Topic is FNC studies. Within 2 minutes, I will join. Okay. Please inform to your friends. Let them join as early as possible. YouTube Live and Facebook Live is also going.
Very good morning, Maheshwari. Can you hear me?
Yeah, your voice is audible, sir.
All right. Let me just share my presentation.
Yeah, okay.
Yeah, are you able to see my slides?
Just a moment. Yeah, I can, sir.
All right. So just let me know if you're ready to start.
Yeah, you can continue, sir.
All right. So welcome all of you on a Tuesday morning for our ongoing lecture series, online training on drug discovery and development. And let's just get on with our lecture. So as usual, we'll do a recap of what we have learned yesterday, and then we'll continue with our new topic. So yesterday we have talked about animal handling, routes of administration, recommended sampling, and today we will talk about target selection, validation of the target, the disease target, and how do we really do a lead optimization, and then we'll talk about few general preclinical studies, including efficacy, toxicology, and ADME studies. And if time permits, we'll just introduce the topic of GLP, and we will then complete GLP tomorrow. So we talked about toxicology yesterday. Toxicos, it is derived from word toxicos, which is a Greek word, and it is concerned with the study of adverse effects of chemicals on a living organisms. We talked about acute toxicity studies testing. So generally, when you want to study the toxicity or safety profile of your drug candidate, you need to do acute toxicity study. That is one way of studying the safety profile. You can do subacute, subchronic, or chronic toxicity studies, and actually it depends on the duration of your treatment in the target species. Say for example antibiotics, so we know the duration is going to be just 3 days or at the most 5 days, so accordingly safety data needs to be generated for same duration of time. Suppose it is going to be an anti-diabetic drug, then you know the treatment is going to be a long-term treatment, so in that case you can plan to do a subchronic or a chronic toxicity studies, or even long-term carcinogenicity studies, or which are reproductive toxicology studies for a year or 2 years. So based on the duration of a treatment in your target species, you can decide whether to go for acute or subacute, subchronic, or chronic toxicity studies. All the regulatory guidelines for different countries, they follow different guidelines, but by and large OECD and ICH guidelines are widely accepted guidelines. India is also an OECD member, so whatever data is generated in Indian CROs as per the OECD principles in a GLP certified lab would be acceptable in all the other OECD countries. Then we talked about we are talking about animals, so we consider them as experimental animal. So any live vertebrate animal used or intended for use in research, testing, or teaching is called as experimental animal. Laboratory animals, they are specially bred for research purpose only. So they are born in lab and they are utilized for research, and then they are sacrificed within the lab. So they practically never are exposed in outside environment. They are always within the laboratory environment, controlled environmental conditions. Why we consider animal or what is an ideal animal model means you can select any model from laboratory animals. What you should look for is the similarity. What is the ability to extrapolate the data from that species to larger species, especially human? Are they reproducible, right? So that across generations you can check the data and the safety and efficacy, availability, how easily they are available, the access to those animal species, size and fecundity, of course smaller the size easy to handle, and shorter lifetime, so that the effects of your candidate drug candidate can be studied across lifetime, right? And they should be inexpensive, of course, because they are more expensive, then it will make your entire project more expensive. So we want to cut short the pricing. So based on the cost, also you can decide your model. We already have detailed in detail, we have discussed about different animals available like mouse, rat, guinea pig, rabbit, and dogs. So the most common strain in mouse is CD1, Swiss albino mice, L. balbci. In rat, it is Wister and SD rat. In guinea pigs, it is Hartley, and New Zealand white rabbits are the most commonly utilized rabbit in preclinical scenario. And we have beagle dogs. And then we do ecotoxicology in Japanese quails, fish, honeybees, daphnia. All these are the most common strains of animal species used in research. Are there any alternatives? Yes, of course, there are alternatives. You can do a cell-based in vitro models, as we have discussed about ADME, about metabolism in particular. You can use cell-based models like hepatocytes or enterocytes to study the PK properties of your drug. So you can screen your molecules by doing in vitro techniques. Wherever possible, so you can use lower species, right? Then there are immunological techniques. Then quantitative QSAR, we are yet to cover that topic. We will discuss in detail about QSAR also. And then there are softwares available which are built up based on mathematical modeling of physiological processes. So even that can give you some idea about the DMPK or safety or efficacy potential of your drug candidate. Okay. And finally, you have human models. Human model, I'm not talking about the live human models, but you can actually use simulation models. And then nowadays, a lot of softwares are available where the actual simulation is created depending on different system, like circulatory system, reproductive system, all those in our human body, same is simulated. So you can use those techniques to screen your molecules, but finally, as we have discussed in detail earlier, we may have to use animals as our testing of drugs, because without animal testing, I don't think drug discovery is possible. You can definitely use alternatives to reduce the usage of animals, but you cannot completely eliminate the role of animals in drug discovery. Then we talked about housing of experimental animals, temperature, humidity, air pressure, different parameters. Everything is well defined in CPCSA guideline. So I would recommend everyone to download that guideline and read the requirements, and then see if your own college, university, lab or animal facility, do they comply with all these standards as mentioned in CPCSA guideline. So temperature 23 plus minus 2 degrees, humidity should be 30 to 70%, light intensity around 350 lux should be the minimum light intensity, around 15 to 20 air exchange, and it should definitely be specific pathogen free. It should be sterile, as we have discussed. It should be class 1 lab facility, and they should, wherever possible, don't keep them in isolation, always keep them in groups so that socializing environment is there. And we had a detailed discussion on enrichment. So wherever possible, please provide them with enrichment so that they are happy, they are more than happy, they're comfortable, and they are like whatever data you generate from that is reproducible and reliable, as long as they are maintained at ACE. And eventually you should maintain good animal welfare practices. Then we talked about animal handling. Avoid lifting them from their tail, and always you should wear the protective ears. The way you should hold them is like the skin between their on their neck, on back side of their neck, between the two ears, with your left hand. If you are a righty, if you're right-handed, then with your left hand you can lift that flap of skin, and then flip it around as in this. And now with your right hand you can actually give the oral intubation tube and give a oral administration of drug, oral gavage, or you can give them an intraperitoneal injection or intramuscular injections. Again, when you try to lift them from the cage itself, please try and make sure that do not be harsh or aggressive. And again, don't lift from tail. Instead, your 3 to 4 fingers should go underneath their abdomen, and thumb should be on the top, going right next to the ear. But make sure that your thumb is slightly behind, because they may turn around and can reach to your thumb, then they may give you a bite. But as long as you handle this way, I'm sure animal rats or mice don't feel any excitement, or they don't really feel aggravated. So they'll be very happy if you handle them like this. And then, as I said, you can then little bit squeeze them with not much pressure, but now they are completely locked and you can really handle them well. Even for larger species like rabbits, just like a baby, we hold from with two hands, one behind their muscles, thigh, and other behind their neck. If like that, if you lift, they'll feel very comfortable. This is another example with a beagle dog, how you lift them. If you treat them like your own baby, they tend to be very calm and cool, and you can do whatever procedures or interventions you intend to do. So then we also discussed about administration, like different routes of administration, and the recommended volumes, blood volume you can withdraw as per the guidelines. So dosing we talked about. Basically, you need to administer the drug. If your intended route of administration in the final species, in your target species, that is human, is oral, then same route you have to also follow in animals also. Suppose if it is intramuscular, then you should do intramuscular studies in animals. And dose volume based on the dose concentration and chemical physical properties of substance and species of the animal, they are well defined as per the guidelines of CPC-SEA. Same is the case for blood volume. So for oral gavage through drinking water in a capsule, you can just see mouse, rat, rabbit, dog. There are different dose volumes mentioned, and accordingly you should treat them. If you give them higher dose volume than it is recommended, then basically it will overload the stomach and it will pass immediately into the small bowel. So it will have less time for digestion, for absorption. So sometimes if there is a larger volume in the stomach, it may reflux back into the esophagus. So you have to make sure that always follow the recommended dose volume of administration as per the species. Parental route, then we talked the factors which really decide the parental route, what you should consider. Dose volume, the stability of the formulation. We already talked about, like if your drug is liable to hydrolysis or degradation via esterase enzyme, then you should make sure that you have done all those studies prior. Otherwise, the moment you give a parenteral dose intravenous, it may get degraded. You should make sure the pH, because it should match somewhere to blood pH. If it is acidic pH, which is again not good, because there will be a big pH difference as soon as it sees the blood. It should be sterile because it is directly getting into the circulation. Otherwise, it can lead to septicemia and toxemia. So it should be biocompatible. The needle size should be as small as possible, and most important is the speed of injection. Yeah. So for subcutaneous route, which is very important for drugs which needs prolonged absorption time, we discussed about the example of insulin. So subcutaneous is the most preferred route of administration. Again, there are dose volumes mentioned as per the species. Higher the species, lesser is the dose volume. Intraperitoneal, I already talked about it. Once you hold the rat or mice by pulling the flap of their skin over the neck, then you flip around. Now the entire abdominal cavity is exposed to you, the peritoneal cavity. You can now easily give the intraperitoneal injection. So you should be careful, as I said. If it's a wrong dosing, it can go directly into the intestinal tract also. So you have to make sure that you pull a small flap of skin, and after putting your syringe needle inside, you have to withdraw a little bit. If any blood comes, means you have intervened any of the organs in between. If there is no blood inside while you withdraw the syringe, means it's a right dosing site. Now you can give the intraperitoneal injection. Intramuscular, of course, the absorption is faster than subcutaneous, and many of the injections are given intramuscular, as you already know. So in muscle, it forms a depot actually, and then over a period of weeks time, it slowly gets released from that. So for that also, species-wise dose volumes are all well defined. Of course, it is 2 less in mouse and up to 250 microliters in dog and rabbit. Intravenous, as I already mentioned, directly into intravenous, and it can be given by 3 ways. One is a bolus injection, which you might have seen that sometimes injections are given. In less than a minute, the entire dosing solution is injected into the intravenous. So that is bolus injection. If slowly it is released into the intravenous route over a period of 5 to 10 minutes, that is called slow intravenous. And sometimes you have to put a drip, and that takes hours together. That is intravenous infusion. Okay, intradermal, again, very less volumes of around hardly 50 to 100 microliters can be used. Okay, and back of the abdomen is the site where you can give intradermal. And the best way to find out whether it is correct or not, once you give the intradermal, basically there are a lot of fur on the skin of the rats and mouse, so you have to do the shaving, and now you can slowly feel that flap of the skin, and you can put that injection inside intradermal. If it is correct intradermal, it will form a small blab with which will confirm that you have given perfectly intradermal, and with time it will subside. So for blood sampling, it totally depends on circulating blood volume in the laboratory animal. So each animal, as it is mentioned, for mouse, rat, rabbit, it is in range of 70s, 60s, 50s, and in dog it is 85 ml per kgs. So as we have discussed yesterday, that although recommended blood volume many a times as per guideline is 10% of the total circulating blood volume in a species, but I would recommend personally that please try to be in the range of 7.5% of the total blood volume you can withdraw, either at single time point or over multiple sampling. But once you withdraw 7.5% of the circulating blood volume, then you have to give them at least 1 week of period to recover. That is called recovery period, within which the lost blood is again made in the body, and now they are fresh again and they are healthy enough to, if you want to reuse the animals, you can use it. So if you are done for 10% blood volume you have withdrawn, then you should keep them for 2 weeks. If it is 15%, you should keep them for 4 weeks for recovery. So the total blood volume we discussed, depending on different species, this is how it is. You should maximum you can collect. So from a 1.8 ml blood of a mouse weighing 25 gram, preferred would be hardly 100 microliter. You see. So that's why whenever you do a PK study in mice, we use them to collect only 1 to 2 blood samples. So instead, different time points we collect from different animals. So we give them sufficient time to recover also going forward, and we don't go beyond 7.5 to 10% of their circulatory blood volume for withdrawal. In a rat of 250 gram, there will be around 16 ml. You can collect up to 1.2 ml. So if it's a repeated sampling, make sure that total blood collection does not exceed 1.2 ml if you want to stay in the range of 7.5% blood volume withdrawal. If it is 10%, then you can still withdraw in total up to 1.6 ml. And generally we use a retroorbital route where blood simply just oozes out, and you tend to collect more than what you anticipated. So you have to be very careful while doing retroorbital. So we always prefer and recommend that you should do by jugular vein cannulation, so that blood withdrawal can be easy, and it can be very precise. You can even take microliters of the blood if you are doing jugular vein cannulation blood collection. Same is for rabbit, and for dog you can collect up to 17 ml or 64 ml if you want to be in the range of 7.5 ml percent. In case if it is more, then you can follow this tabular blood volume withdrawals. Again, it is well defined, mentioned in CPCSA guideline also. So the major sites, the common sites of blood sampling we'll see in this subsequent slides. So for mouse, it is syphinus, lateral tail. For rat, it is syphinus, lateral tail, and sublingual, that is below the tongue. There's a lot of vasculature. So if you can put a glass capillary if you can just prick it below the tongue, there also you can collect few microliters of the blood very easily. For rabbit, it is always the ear, marginal ear or the central ear artery is the main site for blood collection. For dog, again, it can be jugular or cephalic or syphilis. So then we yesterday discussed about toxicity studies, like acute toxicity studies. As I mentioned, the treatment duration, if it is 24 hours, then you intend to do acute toxicity studies. If your treatment duration in the final target species varies between 24 hours to 30 days, you should do subacute toxicity studies, like 28-day tox studies. If your target species treatment falls within the range of 30 to 90 days, then you should do subchronic toxicity studies, typically done for 90 days. And chronic toxicity studies are typically done for 6 months and minimum 6 months. And if your final treatment is more than 90 days, then you should do chronic toxicity studies. Okay. So these are for the acute oral toxicity studies. This is what we have discussed yesterday. Please follow OECD guidelines 420, 423 and 425 and you will see all these protocols well defined in there. Acute oral toxicity studies determine the range of exposure where the lethality is expected, right? Basically, we want to see if at all there are any mortality is observed or not. And then basically you want to do the hazard classification also. And based on this acute oral toxicity study, you can decide the dosage for repeated dose longer studies, maybe subchronic or chronic studies. So repeated dose toxicity studies, as I mentioned, to evaluate the adverse effect as a result of repeated oral administration, which can be a subacute repeated dose, a subchronic repeated dose, or chronic repeated dose toxicity study. In terms of dose level, no matter what toxicity study you are doing, try to have a control, a low dose of your drug candidate, mid dose, high dose, and always try to keep control recovery and high dose recovery groups as well, where that is after you stop the treatment, you want to give some period to the group, recovery group, so that you will understand three phenomena. If there is any toxicity, and if you want to see that after cessation of the treatment, that is when you stop taking the treatment, can those toxicological effect can be reversed? So you will understand that from a recovery group. Secondly, if there are any delayed toxic effects, suppose during the study period, say 28-day tox study, you may not have seen any toxic effect, but after you stop that, over a period of time, say next 14 days, do you see any delayed toxic effect? That is second thing you can study from that. And third and most important is, if there are any toxicity adverse effects, you want to see next, suppose it's a 28-day tox, so in next 14 days when there is no treatment given, you want to see if toxicological effects are still continuing. So these three effects you can study by having two additional recovery groups. One would be for the control, another would be for the high dose. So you don't keep recovery groups for mid and low. Whatever data you get from high dose, you can easily extrapolate that to low and mid dose as well. In general, for any toxicology study, what are the observations we made? Daily basis, you need to do clinical signs observation, like their behavior, clinically, if they sound okay, ophthalmological examinations, all those things visibly, whatever you can see, you should check on a daily basis. Of course, we look for the mortality. If any mortality happens, then immediately it can be taken into consideration. If any morbidity happens, if animal is lethargic, even that can be reported. On a weekly basis, you can do the body weights of the animal, so that data would be available. As I mentioned, you can do ophthalmic examination prior to the treatment with the drug candidate, and after end of the toxicology study, you can do an ophthalmology examination. You can check for food consumptions on a weekly basis. Hematology, clinical chemistry, NBO, FOB, all these things can be done. They are the general endpoint parameters for any toxicology study. So we also talked about minimum space requirement. So based on the size of an animal and number of animals you're going to keep is decided in a cage. So based on the weight, basically, if it is less than below, you can see at least 10 animals per cage you can keep, and the size of the cage of a rat to be taken into consideration is this, right? Same way for the mouse. So based on their body weight, you can decide in the cage of this dimension how many animals can be kept. So basically what we discussed throughout the drug discovery development phase, all these experiments of in vitro, in vivo experiments, IVIVC, screening for cytochrome P450 inhibition, induction assays, definitive in vivo assays, ADME, all this thing goes on in different phases of drug discovery, and sometimes they overlap. Say for example, IVIVC, it actually overlaps between discovery and preclinical development. Same way ADME studies, it overlaps between preclinical and phase 1. So like that, you have to keep doing all these DMPK studies throughout drug discovery and development, at least up to phase 2. So basically, in a drug discovery development process, you do refinement of your chemicals, like the drug candidates, similar scaffolds, hundred thousands of molecules which has come to you. You do that screening of molecules, and then you do chemical biological characterizations. Then you do safety toxicity studies. Finally, they are tested in human, and then after regulatory process, it gets launched, marketed. Post marketing also, you look for any adverse effects or any learnings in terms of lack of efficacy or something, and you can learn your lessons and then further develop your molecule, right? So in nutshell, what we call as discovery means to find a new active structure, and development means now you have found a structure by optimizing the DMPK properties, by optimizing the formulation, by optimizing the efficacy toxicology of to be elicited by that drug. You are basically making that active structure in converting it into a drug. So conversion of that into a useful drug is called development. All right. In a nutshell, whenever you do an in vitro animal experimentations, basically in vitro metabolism studies, you try to extrapolate the data in for an in vivo study, or you can actually do an in vivo animal study also, and you can do an in vitro experiments in human. So you've got in vitro animal data, you have in vitro human data, and you have in vivo animal data. Using all these three information, you can easily predict the clinical outcome of a drug in terms of its efficacy, safety, or most importantly, PK properties. So that is the role of in vitro in vivo correlation from preclinical to clinical. So let's now jump onto our today's further discussion. So today we will quickly touch base on drug discovery development process, which we have talked about it in our initial couple of lectures, but in just 2 or 3 slides. And then we will, because if you want to discuss about target selection, actually it starts with it's the first step in drug discovery and development. That's why we need to touch base on that, and then we get into target selection, validation, and then so on. Okay, so let's understand drug discovery development process and where does the target selection actually lies. So you see, if you look at this diagram, this will really, I will spend just a couple of minutes over here. So you know, the first and foremost step in a drug discovery is identification of a target. What is that? That's a disease target. So once you identify a disease target, basically it's nothing but you have to find the mechanism of that particular disease. If you can crack that mechanism of that disease, then it's very easy to identify a target whereby you can intervene that mechanism, disease mechanism, so that to elicit a pharmacological action over it. It could be inhibition, it could be induction or activation or deactivation of any of the pathways, but that is the first step called as target selection. Correct? So to understand that, you need to do molecular studies. So basically, as I was mentioning, eventually the target is going to be a molecular target. It could be a receptor, it could be an ion channel, it could be a transporter, it could be an enzyme, or it can be a signaling molecule, correct? So your target is mostly is going to be a molecular target, correct? So first and foremost thing is to identify the disease, to understand the underlying mechanism of the disease, and to find out that molecular target which if you do an intervention of that will definitely impact the mechanism of disease. So either it will stop the disease or prevent the disease, or it will treat your disease. So that is called molecular target selection, okay? And once you have the target, you actually you need to validate that. It's just that not only by literature that okay fine, this particular receptor is activated, then this pathway is going to be impacted and finally the disease would be cured. So you can study all this thing in theory, there's a lot of literature available, but that doesn't really serve the purpose for drug discovery and development. First you have a literature review, research, whatever you want to do, but finally you have to actually validate the target. Suppose if I feel a particular ion channel, if sodium channels or a chloride or whatever ion channels I'm working on, calcium or something, if if I do intervention of that, it's going to have a cardiovascular protective effect, but I'll have to prove it. Yeah. So you need to develop an assay. First of all, you need an assay. Now, assay could be in two forms. It could be a cell-based assay or an enzyme-based assay. So if your molecular target is an enzyme, then you can have an enzyme-based assay or a cell-based assay where your target, molecular target, should be expressed. Okay. So that now you have an in vitro tool to test if at all your drug can be tested for positive impact or not. In the sense, now you have an in vitro assay in place where you can screen a series of molecules if at all they are able to inhibit or activate or induce your molecular target which is expressed in a cell system or an enzyme system. So that is called development of an assay, correct? So now chemically diverse molecules can be screened and you can see if at all you hits out of it, correct? So called development of an assay. As it is said, you can always use a automation, use a liquid handling units, and in a 96 well plate or a 384 well plates if you use, if you develop an assay, then it is a high throughput fashion, right? As many wells can be utilized to screen your as many number of molecules. So that is the way of doing lead search basically. So from that diverse chemical structures what you have tested, you will come up with few structures which will show you good result in your lead search. So once you have that thing identified, we call that as lead, and now those lead molecules gets into optimization. So what is that optimization? As I was mentioning about ion channel or say a specific receptor, we want your drug to be highly selective to those receptors or ion channels. Suppose if my drug is supposed to act on sodium channels, then I don't want it to act on calcium channels, right? I mean, just an example, so that it is highly selective, because if it is highly selective, then it is more safer, right? If it is not selective, it can hit any other target and it can elicit some effect which may not be desired. So from a safety perspective, we want to check if it is selective. So in case if it is not selective, what does happen? What you can do? So that is lead optimization. If in case it is not selective, you can go back to the chemist and request him to modify the structure slightly so as to enhance the selectivity. Same is the case with efficacy. Efficacy generally comes under optimization. It needs to be done in an animal model. Say for example, diabetic drug I'm working on. Then I want diabetic, there are two ways. I can either induce diabetes like streptozotocin, alloxan induced diabetic models, or I can use genetic diabetic models. What are those? Like Zuckerpfeffer rats, DBDB mice. So these are all genetically proven, they are genetic models where they exhibit high glucose levels, right? So you can use such models and use your drug and make sure that eventually it brings down the glucose, or it activates the release of insulin, thereby it is efficacious. So that is the efficacy you have to check in animal model based on disease. If it is oncology, you need to have a tumor model. As we have discussed yesterday, you have to develop a tumor model in a skid or a nude mice and test your drug for the efficacy. You also have to study the tolerability. So tolerability is nothing but you want to see how much maximum dose it can accept. So that is tolerability. You have to do pharmacokinetics because we want AUCs, half-life, Cmax, concentration maximum, Tmax, clearance, volume of distribution. We want that data. So you have to do a pharmacokinetic studies in animals. So this is all the process which we do in lead optimization. As for efficacy, as I was mentioning, you have to use a relevant species, like you can use a knockout or a knock-in mice. Right nowadays, customized genetic models are available based on the disease target you want to work on. You can use an agonist or antagonist, antibodies. All these things is possible using animal studies, especially for efficacy study. So all that thing what we are doing, trying to optimize our lead, is called lead optimization. So please remember, at any point in time, whenever you feel there is any issues related to efficacy or tolerability or metabolism, you can always go back to chemist as long as it is in lead optimization stage and play around with the structure. Somehow, if structurally you can fix those issues. If not, if yes, well and good. If not, then we try to concentrate on formulation, and somehow we want to see if by putting our efforts in formulation development, can we address any of these issues, especially issues like solubility, permeability. All that thing can be solved using formulation efforts. Okay, so that's about lead optimization. So after all that, then you come up with one drug candidate which is really optimized lead, we call, and then you want to do the safety testing for that, right? Based on, as I mentioned, based on the duration of treatment in the target species, you can decide the toxicity studies. So you can do an acute, subacute, chronic, or subchronic study based on the final duration in the target species. And then we go to the human clinical trials, phase 1, 2, 3, and then it gets approved. So you see, this entire process of drug discovery development basically starts with identifying the target, actually developing an assay which expresses the target, and then testing your drug to find the leads out of it. And once you have the lead, lot of efforts goes into lead optimization, which includes a lot of in vivo experimentation. All right. So just to summarize this, target identification and validation. Basically, a molecular target has to be proposed, it has to be identified, a biological hypothesis relevant to the disease you have to come up with, and then you have to check in a genetic models to demonstrate the proof of concept that in a live system, does it really happen? So you can use a knockout or a knock-in mice, rat, and then demonstrate the proof of concept. As I mentioned, once you have a biological assay, you can do high throughput screening of your compound libraries. So this can be even done in silico. Basically, you don't need a in vitro assay. So as I mentioned, do in silico, then the confirm potency and selectivity from the hits, and then finally go to in vitro PK assessment in microsomes or hepatocytes. Lead optimization, as I said, a lot of efforts goes from chemistry to convert those few hits into leads. So always have an assay in place where you can check the potency, selectivity, also look for liabilities of CYP inhibition, metabolic stability, and then do an in vivo efficacy in a relevant disease model, as we have taken the example of diabetes. So that's how you develop a lead optimization. So a lot of extensive medicinal chemistry effort goes into, because if you say it is not selective, medicinal chemist will have to do some alteration in the structure. If you say potency is not that great, then again he has to work on his structure. And then you do an in vivo efficacy study in additional models if it is available, to get a more confidence on the potency of your drug. Do entire PK characterization, and then you find out initial dose ranging studies for tox studies, and finally it comes to the stage of preclinical development, where You do GLP compliant toxicology studies, like all the acute to subacute chronic studies are done in a GLP compliant manner. You have to do the safety studies as per the GLP compliance. All the formulation studies needs to be done, and with all this data, whatever data you generated here, that gets into your application of IND filing, right? Investigation new drug filing. You have to include all that data, majorly the GLP data which you have generated for preclinical development. Okay, so that's about target selection and validations. As I mentioned, starts with defining the unmet medical need, understanding the molecular mechanism, identifying the therapeutic target. As I said, could be ion chain, receptor, enzyme, or a nuclear receptors, and then you have to demonstrate that target is relevant to the disease mechanism. Okay, using animal models, compounds, or relevant antibodies and all. So once you have that in place, you need an in vitro assay. Then you can do an in vivo proof of concept study. You can do in silico screening first, followed by in vitro, and then followed by ADMA screening and in vivo efficacy study. Finally, your drug candidate is ready to go for safety assessment. And once you do that, now your preclinical candidate is narrowed down, it is in place. So that's why I always say the first step, the most important step in drug discovery development is identifying the drug target. Okay, so again, it's just the same thing. Just remember, develop and validate robust biological assays, either in a whole cell or a transformed cell or a cell-free biochemical assays also. Okay, and nowadays a little bit on high throughput, because the hit, like chemically diverse compounds you can test, but you have to make sure that whatever cell based or a non cell based assay you're using, it can be done in high throughput fashion, right? State of art technologies can be used, so that they are done in a high throughput fashion. Correct, I am talking about in vitro assay, but there are compound libraries when you are screening. Okay, that can also be done in a high throughput fashion. Correct. So starts with hits and create the lead compounds. So what is the job of a chemist? Basically, he will synthesize and test the analogs. Suppose you have a lead, then he will further synthesize the lead-like compounds and all the analogs to basically optimize the properties. Which are those properties which are important from a chemistry perspective? Potency. For off-target effect, it is selectivity, and related or unrelated targets to avoid the side effects. So drug design to create the novel compounds, basically nowadays it is being used as computer-aided drug designing. Okay, so we'll talk about it separately also, what are the softwares available. But from Infosys and all, there's so many, from Dell and all, there's so many systems available, so many softwares in place. Using that, you can do computer-aided drug designing, right? So okay, so novel compounds could result into variable intellectual properties. Yes, of course, if they are novel, you will have a scope to file for a patent. In vitro ADMA characterization is very important. That is lead optimization. It happens during that stage where you want to address solubility, permeability, metabolism, and excretion of your molecule. Even the proof of concept, initial proof of concept, can be a non-GLP studies, but finally you would like to do a study in a efficacy model, genetic model also going forward. So other way of finding the leads is you have an active compound already identified, right? You can modify that, and then like in this case, they have taken an example of 5-hydroxytryptamine. So you see, it's a serotonin, a natural neurotransmitter in the central neuron in central nervous system. It is a neurotransmitter. It is synthesized there. So this is the structure of that. Now, after a lot of chemistry efforts, if this hydroxyl site, if you modify an SO2 and amine group is added here, you see now it has become sumatriptan. So now this can be used. This is actually used to treat the migraine, because it is 5-HT1 agonist. This is a structure of serotonin, and this is an agonist of that. So just you see, by modifying the existing active compound structure also, you can come up with new compounds or new leads. Okay, so what does it basically do? It improves the biological activity and it improves the selectivity of the target also. Okay, so it improves the therapeutic index also, and maybe improvement in the physicochemical properties. So now it's a 5-HT1 agonist, and it is marketed as Imitrax sumatriptan, right, with modifications on hydroxyl group of a 5-hydroxytryptamine, which is a known active structure of serotonin. So, as I mentioned, you also want to do in lead optimization. You want to check whether the synthetic scalability is there or not. Like so far, a chemist must be synthesizing few mgs of drug, but you want to check during the lead optimization, can this be scale up from mgs? Can you go to grams, or from grams to kgs? If that is doable, fantastic. If it is not, then that can also be one of the challenge, because no matter how good a drug is, how potent, how safe a drug is, but if you cannot scale up the synthesis process, it can never come into the market, right? So we try to increase the in vitro potency as much as possible. If at all there are any off-target effect, so by chemistry effort during a lead optimization stage, we would like to increase the selectivity. Of course, in vivo efficacy also gets in there. Entire PK, as I already mentioned, half-life, Cmax, Tmax, AUC, clearance, everything needs to be optimized, characterized, and optimal mode of delivery. This formulation development also has to happen. So basically, lead optimization is amalgamation of so many different activities. So interdisciplinary approach is required for lead optimization. So it's a collaborative work of pharmacology, toxicology, PK scientist, biology, process chemistry, and even regulatory people from who have knowledge on patent filing and all. So you see, it's a collaborative work between different disciplines. So what is a summary of lead optimization? Let's understand. You want to tend to check, let's start from here. When you do a target selection, most of the genomic knowledge, genetic target is going to be, so that comes into the play. You want to develop, use a software and then try to find the hits. From hits to lead. So as I am saying, it's using software, so there are libraries available. You can look for HTS, and from that, once it gets into lead optimization, you want to look at the scalability, how easily it can be scaled up in manufacturing. You want to look for in vitro selectivity. You want to optimize in vitro metabolism. You want to check the proof of concept in animal model study, and you want to check for in vitro toxicity studies, which we will talk about it later, like AIMS, mutagenicity, micronucleus test. All this thing can be done, and then finally only 2 to 4 compounds will pass this phase and they enter into development phase. So you see, a lot of lead optimization effort goes in, and different disciplines work together to progress the compound from early discovery to development phase. And in development phase, as I mentioned, it is all done as per the GLP OECD principles and guidelines, and definitely in a GLP compliant manner. So we call it as GLP toxicity studies. And once we have that safety study in place, then with all this data we apply for IND application for permission for FIH, which is first in human, which is called phase 1. So that is the important role of lead optimization in drug discovery and development, and about target selection and validation. So now, if we revisit this particular funnel of drug discovery and development, you will realize that thousands of molecules through compound libraries are screened using in silico methods for hit identification, and then from hit to lead, and then lead optimization. So very few compound will enter into preclinical development, and hardly we can count on fingertips that many gets into clinical trials, phase 1, 2, or 3. Phase 1 is always healthy volunteers, 20 to 100 in numbers, followed by phase 2, where 100 to 500 volunteers, which are essentially patients, and then phase 3 are large scale multi-site clinical trials in patient population for about 1,000 to 5,000 people or even more. And then finally you submit NDA based on the clinical trial, and then they review and then they approve the drug. So that is the whole process about drug discovery and development, and that's why it takes a lot of time for each of this phase. So that's why we say that more than two or billions of dollars of investment, and in total around 12 to 15 years it takes for complete drug discovery and development. All right, so IND, investigational new drug, GXP, because you can replace X with L, it becomes GLP, good laboratory practice. You replace it with C, it is called good clinical practice. You replace X with M, then it becomes good manufacturing practice. So some FDA expectations, and let's understand through some examples. So investigational new drug application. So the primary goal, as we have seen, is to present the data. Okay, so the data package is being submitted to FDA, okay, to allow the initiation of a clinical program. So basically, we have all the data from early drug discovery and preclinical GLP safety data, efficacy data, PK data. We submit this data package to FDA, and basically we ask the permission that please allow us, based on this data, once you're convinced that it is safe, it is efficacious, and it is optimal in terms of DMPK so properties, so we should be given permission to conduct first in human dose, which is clinical phase 1 trial. So that is the purpose of IND filing. Okay, so to present the data to justify that compound exhibit pharmacological activity to meet an unmet need, correct? Because our first step itself was to identify an unmet clinical need, that is to identify a disease target, and then to support the product being reasonably safe, because you have now all the preclinical safety data with you, so that it is reasonably safe for initial use in human. Okay, and to justify exposing human to reasonable risk when used in limited early stage clinical studies, because eventually we are going to do clinical trials. So through this data we want to justify that there will be reasonably less risk if we have to use it in clinical trials. So after reviewing these three things, they really either have queries, they come back to you, or they reject your application, or they approve you to conduct IND to conduct first in human clinical trial. So as I mentioned, GXP, if you replace X with M, that is GMP. So an IND application must contain information in three broad areas, as I mentioned. Manufacturing information, so that would be good manufacturing practice, which information should include the composition, right? The manufacturer, the stability data, and controls used for manufacturing the drug substance and the drug product. So this information is assessed to ensure that company can actually adequately produce and supply consistent batches of the drug. As I mentioned, if there is no scalability potential for a particular drug scaffold or a chemical structure, then it cannot proceed further. So that IND will have a lot of questions. So you have to show the scalability, and you have to have the stability data, because for whatever period it's going to be, say you can do an accelerated stability studies, or you can do a long-term stability studies. So either way, you have to submit the stability data. Then they also look for, in IND application, they look for preclinical data to permit an assessment as to whether the product is safe, because in preclinical species what we have done, we have checked the efficacy, we have checked the toxicology. So if it is reasonably safe for initial testing in human or not, so it requires animal pharmacology and toxicology data. Generally it should be in a GLP compliant lab, good laboratory practice certified labs. Okay, and then we want that clinical protocols and investigators information as well, because you want to go in human, but you need to submit a detailed protocol for proposed clinical study, where you will access the initial phase trial with exposure, expose subjects to unnecessary risk, means we want to justify that there is no exposure to initial healthy volunteers to any unnecessary risk. So you have to share that detailed protocol. Also the information on qualification of a clinical investigators, because who are the professionals going to be involved in clinical trial, clinical investigator, principal investigator, the site investigator, all those information you have to mention, because who is going to oversee the clinical trial also. So finally, the commitments to obtain informed consent from the research subjects to obtain the review of study by an institutional review board and adhere to the investigational new drug regulation. So this three basically information is what an IND application asked for. I mean, the regulatory agency asked for. So, in your IND application, if you have manufacturing information, if you have animal pharmacology and toxicology studies data, and if you have clinical protocols and investigator information, then it is complete data set for them to review and give further permission or questions to you whether your drug is ready to go in first in human trial or not. Down is what mentioned the guideline. You can definitely check www.fda.government site. There you can look for approval process and the data investigation new drug application requirements. So there is a detailed requirement of what all things you basically need to do the IND filing. All right. So when you say GXP criteria, basically quality systems and requirements, right? So there are two words, quality control and quality assurance. So you should have a quality control, more so from a manufacturing point of view, and you should have a quality assurance more so from a GLP perspective, from a pharmacology or toxicology study perspective. You should have these two things in place. Why? Because if you have quality control and quality assurance, you can ensure the uniformity, right? Consistency, reliability, quality, and integrity of the product manufacturing, toxicology, and clinical trial conduct. So that is the importance of quality systems, right? So you should have quality control and quality assurance rightly intervene within the institute wherever you are doing either preclinical development or you intend to do the clinical trial or even the CMC efforts. So what are those IND enabling preclinical studies? So basically efficacy studies, yeah. So in animal models you need to do safety pharmacology studies, general toxicology, which can be oral, could be one of the route, and specific route of administration. If it is not oral in the target species, then you can use that specific route of administration. You should have a genetic toxicology data, PK data, of course, local as well as systemic. Why? Because we want to know the bioavailability. So after systemic administration by intravenous, and one local, which could be either oral, intramuscular, or any other intended route of administration in your target species. So you should have a complete data package to understand absorption, distribution, metabolism, excretion. You should have reproductive toxicology data, carcinogenicity data, and then if at all some special studies are required, that. So this entire data package is prepared for IND submission, right? So why IND enabling studies are needed, right? So basically FDA will determine during pre-IND meeting. So before actually you go for an IND filing, you can do an pre-IND meeting, right? So that will really decide what all things you need to really submit in particular. Otherwise, of course, there is a detailed data package what you need to be ready with. But after this pre-IND meeting, it can be decided. So basically, what they look for is what is your patient population, right? What are the patient? What is the disease target? What is that unmet medical need? What is the expected dosing? Is it for short-term duration or a long-term duration treatment? How it is going to be delivered? That is route of administration. Is it by oral or IV? So based on these answers, relevant data would be required. So as I mentioned, you have to demonstrate safety preclinically. Only then you get a approval for first in human study. Got it? So yes, this is just a cartoon to, I mean, just to divert slightly, that the people are sitting and they're discussing, should the disclaimer for our client's new sleeping pill, should it read may cause drowsiness or may not cause drowsiness, right? So this is just cartoon to divert you slightly from the very serious discussion what we are having on IND enabling studies, right? So this is about the label for a drug, what the discussion is going on, right? And as you may see, there are 50% chances, right? It may cause drowsiness, it may not cause drowsiness. So purely from a commercial purpose, you can really decide which one goes well, right? From a commercial angle, if I had to see, of course, it would be may not cause drowsiness. But from a label point of view, I think it should be may cause drowsiness. Okay. All right. So moving on about pharmacology. So in vivo animal models are basically used to demonstrate the efficacy. As I mentioned, taken one example of diabetes, you can use a streptozotocin induced diabetic model. You can have alloxan induced diabetic model, or you can have a genetic models like Zucker rats or DBDB mice. All these are models specific to diabetes. Like that, for each species, each disease, they can be different models, right? So basically, you understanding the pharmacology impacts and the interpretation of toxicology studies is very important in pharmacology, right? So efficacy studies are conducted more for candidate selection and prioritization, correct? So if you have few candidates, just by using their efficacy study, we can make out which one is more active, which one is more potent, and based on that you can rank order those different compounds and take the best molecule further for further testing, right? Safety pharmacology, basically it investigates potential undesirable PD effects like pharmacodynamics. As we know, effect of drug on body, so undesirable pharmacodynamic effects on physiological functions of vital organs. So that is must when you submit an IND application. So generally we say that you have to do a core battery test, and the thumb rule is always try to do it at a dose which is higher than the indicated dose, your oral dose. And core battery test comprises of three systems, major systems. You have to address cardiovascular system, respiratory system, and central nervous system. So these are the three different systems and most vital systems which you have to address when you do safety pharmacology. When you say general toxicology, basically we already talked about toxicology, so you will know that determining whether the proposed clinical trial is going to be safe or not, that you come to know from this general toxicology, right? So what is the main goal? Identify the toxicities, right? Direct monitoring in the clinic, and to calculate the safety margins. So these are the things which you have to do. So what are the important factors? Species selection. So generally one rodent, one non-rodent. So one rat or mice and another should be a non-rodent. Dose selection is very important. So as I already discussed, you can do an acute dose and select the dose for your subsequent study. Duration of administration is important. If it is repeated administration, how long? As we have already discussed, you should decide based on the treatment duration in the target species. What should be the duration of administration in your preclinical species. Route of administration, again the final route of administration in your intended species should be same replicated in animals also. And about the endpoints, we already talked about the detailed endpoints for a toxicology study, be it clinical observations, body weight, food intake, clinical, biochemical, mortality, morbidity, all these parameters, ophthalmological examination, this can be the endpoint of your toxicology study. So let's talk about species selection. I already mentioned two species, one typically rodent, okay, and one should be a non-rodent. So metabolic profile should be similar to human. This is very important. One of the person has asked a question some time back that animals are different than human. Then here is the answer that yes, of course we do safety studies first in animals before going to human, but try and pick a species at least which has shown you similar metabolic profile as that in human. How do you come to know the metabolic profile? By doing in vitro experiments of metabolism using hepatocytes or even enterocytes. So you will know which particular species is exhibiting same metabolic profile as it's intended in human, right? Because you can use human hepatocytes also. So based on that, please select that particular rodent species. So whatever efficacy or a safety or a toxic effect or adverse effect may be anticipated can be reproduced in an animal species if their metabolic profile is same, correct? Dose selection, maximum, it should be up to maximum tolerated dose. So before doing this, you'll be doing a dose range finding studies where you keep on increasing the dose till you reach maximum tolerable dose. So it should be up to the maximum tolerable dose. You should also include a dose where there is no adverse effect level. So the lowest dose try and include in such a way that at that dose there are no adverse effect level observed, and the highest dose you are going to include maximum tolerable dose, and they should be spaced. This low, mid and high dose should be spaced so that there is a complete dose response assessment or a range, correct? So one dose where no adverse effect level is observed, one dose where maximum tolerable is the maximum tolerable dose. So and the third dose should be somewhere in between. So this is how you do the selection of the dose. Dosing should not be determined mere by multiplying the human dose. Very nice, because see, sometimes I see students doing their dissertation work and they simply take up the dose and multiply the human dose just to get a logarithmic exposure. That's not the criteria. Okay, dosing regimen can be adjusted to mimic the human exposure. If TF is shorter in animals than humans, such that exposure with daily dose is limited, give multiple times per day. So this is again interesting. Try to mimic exactly what is going to happen in human. So suppose if TF is shorter in animals, right, compared to what you find in human. So what you do repeated dosing in such a way that multiple dose can be given in a day, but the exposure level should be more or less same what you anticipate in human, so that the data you generate out of this toxic study would be more relevant. So duration, as I said, up to 2 weeks, initial clinical trial up to 2 weeks. Suppose you are planning to do for 2 weeks, then toxicology data is also required for 2 weeks. Okay. For later clinical trial, it must be equal duration. So as I already mentioned that if it is for a diabetic, then it has to be a long duration. If it is for antibiotic, then definitely it's going to be a shorter. So it can be managed sequentially if preclinical is run staggered with clinical, and you should also discuss with FDA. So of course, case to case basis, duration of dosing can vary, but by and large, just keep this in mind. If in your target species duration of the treatment is less, then try to keep same in your animal species while doing toxicology studies. If that is more, then accordingly try to mimic that also in animal species. At the same time, the exposure levels we want to have the same. So if at all the half-life is shorter in animal species, if you know by doing in vitro assays, then you can even do multiple dosing, okay, just to achieve the same concentration or the same amount of drug in the body as it's anticipated in human. So that's about dose selection, about duration of the dosing, route of administration, as simple as that. Please try and keep same as it is going to be in your final species. So typical repeated dose toxicity studies, this is how it looks like. You treat your rat, example rats or mice or whatever your species is, preferred species, and then one of the important thing which is a parameter is the drug analysis. When you take the blood sample, you can find out the you can quantify the drug in that and even the metabolites in that. As I already mentioned, always look for daily clinical signs. On a weekly basis, you should look for body weight. You should look for functional observational battery. You finally, when you sacrifice at the end of the toxicity studies, be it 28-day, 90-day toxicity study or a 6-month toxicity study, finally you sacrifice them. And before sacrifice, you collect the blood. In that blood, you can do clinical hematology or biochemistry. And once you sacrifice, as bare minimal, again it can vary based on the disease target you're working on, but by and large, as per the guideline, minimum 40 organs is what we are collecting from one rat or mice species after sacrifice. And each of those organs can be stored in 10% formalin, and then you can make a slice out of it, undergo the histopathology, and then you can study the histopathological impact also. So in general, typical repeated dose toxicity study design, this many parameters are there to look for. Okay, and all of them should be as per the GLP compliance, right? So it should, your lab should be a GLP certified lab, and the study should be as per the OECD principles of or guidelines for toxicity. So it should be GLP compliant. All right. So very quickly, the endpoints we have covered this: mortality, whether animal is alive or dead. That should be one of the endpoint. Clinical signs on a daily basis, body weight, temperature, activity levels on a weekly basis, hematology, clinical biochemistry at the end of the experiment, while you're going to sacrifice. Just before that, you collect the blood. And toxicokinetics, you can have one satellite group. Suppose I got 4 groups, 1 control and 3 dose groups, different dose groups for toxicity study, and a couple of recovery groups. I should have one additional satellite group where I can do toxicokinetic studies. Finally, after sacrifice, you collect all the tissues and make try to study the pathology, that is histopathology. Okay. So for genetic toxicology, core battery of tests, as I mentioned, if time permits, maybe in subsequent class we'll try to get into the protocols. But for now, just remember the names: microbial mutation test, which is also called as Ames test, right? In vitro mammalian chromosomal aberration test, or in vivo micronucleus test. So these are all the tests which can be done as a part of genetic toxicity. So Ames and in vitro mammalian chromosomal aberration test, they are submitted with initial IND application. Please remember. So it is a must. And the last one is to be submitted before the phase 2. So after this 2, you submit IND, you get a permission to do first in human, go for that clinical trial. And before you start your phase 2 clinical trial, you should have completed in vivo micronucleus test. Okay. So positive signal may lead to additional testing. Suppose if it comes positive, Ames positive, then you have to do additional tests to ensure the safety. Or sometimes you may have to even drop the molecule initially if they are Ames positive. Okay, and sometimes you can, if it is an unmet medical need, and suppose if it is allowed to take it further, you have to have a label which conveys that it is having this potential side effects. From a pharmacokinetic perspective, please remember the parameters under the curve. The most important one which you get from summation of trapezoids, all the trapezoids which comes under a graph which is plotted as time versus concentration, either plasma or serum concentration. So whatever graph you draw, once you draw straight lines which touches the axis, trapezoids are formed. If you find out the area of each of those trapezoids and you sum that, you get area under the curve. Basically, it gives you an exposure comparison under different drugs or different species if you have AUCs there. And as you all know, AUC is also used in calculation of viability, that is amount of drug available at site of action. Actual amount of drug available at site of action is what is called as viability. So to calculate that also you need AUC. Cmax, of course, concentration, maximum concentration which is attained, and Tmax, the time at which after administration of the drug, time at which you get the maximum concentration, that time is called Tmax. We also have learned about T half, clearance, and volume of distribution. So these are all the important parameters which we should study initially. So exposure parameter allow comparison to be made with clinical data so that safety margins can be calculated. So these AUCs basically can be extrapolated to human, and you can have a rough idea about what are going to be the exposures in human. So this will give you confidence on the safety margin if you extrapolate the AUC data from mouse or rat or to other species to human. Okay, so single or repeated dose studies to address the accumulation and induction of metabolism. Yes, of course, when you do a repeated dose studies also for pharmacokinetics, you can actually find out if there is any accumulation in the organs. You can do tissue distribution studies, and there are ICH S3A and ICH S3B guidelines. So one of them talks about toxicokinetics and another talks about the tissue distribution studies. So please refer ICH S3A and S3B guidelines, where it is well defined how to study the tissue distribution also, okay, or toxicokinetics. In terms of reproductive toxicology, basically fertility and early embryonic development you want to capture data on. Embryo fetal development you want to see the impact of drug, or impact of drug on peri or postnatal development. So basically these three types of studies are qualifying as reproductive toxicology. So combined studies are acceptable but require complex design. You can do that starting from say from embryo development stage, from while the last trimester or second trimester is going on. You can start the studies and then continue till the embryo fetal development and all those things, but that designing will be very complex. So it's better to define separate studies for to understand the separate parameters. Okay, and you can also see this is conveyed in the label also, right, that it is schedule H drug, cannot be used in pregnant women, something like that. So this data is very important. Okay, carcinogenicity for any drug where there is a usage of 6 months or more, you are supposed to do carcinogenicity study, right? So or even intermittent use up to 6 months, then also you need to use in your target species, then also you need to do carcinogenicity study in very early. Perform for drugs for which there is a concern based on so that is based on the treatment duration. You can decide whether to do carcinogenicity study or not. And second is based on the chemical structure. Nowadays, with advent of our understanding on chemical structures or the liabilities on a chemical structure or a scaffold which is responsible for eliciting carcinogenic effects, we know whether this drug has a potential to have any carcinogenicity going forward. So based on the drug class, structure activity relationship, pre-neoplastic lesions identified in the tox studies, or the long-term tissue retention studies, can based on this also you can decide if long-term carcinogenicity studies are required or not. Okay, so if you just go by FDA guideline, what FDA site which I mentioned, www.fda.government on the drugs tab, you will see all this detailed documents are there. So in your free time, you can really get access to all these things like investigational new drug application, IND, NDA application for FDA approval, orphan drugs, GLP for non-clinical animal studies, protection of human subjects, institutional review boards, drug labeling. So all this 21 CFR part 312, 314, 316, 58, 50, 56, they are uploaded there. So you can simply download and read if you want to have a detailed understanding on all these parameters on a FDA site, basically for IND submission requirements in preclinical or a clinical development stage. Okay, then of course there's something called as serendipity, a chance occurrence that be accompanied by an experimentalist. Of course, you cannot plan your discovery by chance, as we have seen in the first lecture itself. But sometimes it happens by chance, like discovery of penicillin, which at first place was never intended way of doing discovery. It was a contamination found by chance, and then finally it came out to be a penicillin, right? It was developed as penicillin. So sometimes chance discovery is also there, and that is called serendipity. Okay, so in nutshell, This particular slide will give you an idea uh, about entire process of drug discovery development and where exactly non-GLP studies are there, and from where on all the studies are becoming GLP studies. So all the discovery, screening, lead optimization can be in a non-GLP format. What does it mean? The in silico screening which you are doing, in vitro biological assays which you have developed, all kind of hepatotoxicity, myotoxicity, or HERG for a cardiac effects, HERG channel, whichever you're doing in early drug discovery, are non-GLP. Even AIMS, in vitro collaborations, Comet assay, all this thing can be in vitro assays of genotoxicity can be non-GLP. Even 4 to 14 days dose range find dose proportionality, zebrafish models, all these things are exploratory studies and they qualify as non-GLP studies. So that data is fine for submission. But whenever it comes to regulatory thing, within preclinical, whenever you do a tox study, okay, an acute tox study, acute or say 28-day tox study, or you do an AIMS assay, core battery assay, all this thing has to be done in a GLP fashion, right? When you do a general toxicity, whatever 3, 6, 9 months toxicity studies, all of them are regulatory submission studies. So they are being GLP studies. In vivo micronucleus or DRF, all this thing. You see, chronic toxicity studies, immunotoxicity studies, carcinogenicity, which are done for 2 years time or something for 18 months, right? So all these studies are in a done to be done in a GLP compliant manner, and this is the phase. Like during preclinical, all this thing is done. During phase 1 is going on, you can still continue doing your this data, right? And you can submit, and then you get a permission for phase 2. While phase 2 is there, you can still continue the chronic toxicity study and all the studies like reproductive toxicity studies, and phase 3 and all. So this is the durations when basically you can continue all the toxicity studies during different phases of drug discovery and development. So decision gates in drug development, there are different decision gates in drug development. As you once you select a target, okay, the first and foremost decision would be, is this target validated? Okay, if it is valid, if it is possible to validate, then only it can go further. So it's a decision gate, yes. When you go for lead optimization, I mean hits to lead identification, you have to see, is lead identified? I mean, are you able to get a lead compound from that? So that could be a decision gate, whether you should continue with a particular chemical moiety or a scaffold. So that is a decision maker. When you do a lead optimization, finally the question is, are you able to develop that lead molecule, right, in terms of its pharmacology, toxicology, pharmacokinetics, and a formulation perspective? Then only you can proceed further, right? Same way when you go for preclinical species, are you able to establish sufficient safety data in order to justify the usage? I mean, justify the first in human studies. Only then it can go further. So, like that, at each stage, you know, at even at a clinical stage, does the drug work in human after phase 1, phase 2? You can always question that, and that proof of concept study data will really decide whether further clinical optimization of the dose is required for phase 3 or not, or you can go back and again develop your molecule, you see. And once it is approved, I mean, once you are far end at the clinical trial, it is equally important to find out whether clinical product manufacturing is there in place or not, correct? Finally, whether the drug is approved or not, correct, based on the clinical data, and then the product can be launched. Equally important is the manufacturing process in place, approved manufacturing GMP process in place, and then you can do a post marketing surveillance once the product is launched. After it is being launched, again you have to see whether you're able to sell the drug. There could be so many reasons, purely from a reason of lack of efficacy, for any adverse effect seen after the launch of the drug. You can always go back and optimize the drug again, and then go back to your clinical phase again. So you see, this is also a decision gate. So these are the different decision gates, starting from all the way from your target selection to target validation to lead optimization to preclinical development to clinical development to your final phase 3 clinical trial to the approval from the regulatory agency, and finally after post-marketing surveillance. So at different stages, drug can be dropped or drug can be successful. So just one thing we always say in drug discovery, that if drug has to fail, we would want we would want a condition where a drug fails early. Okay, if drug fails early, it fails cheap. So fail early, fail cheap. If it fails down the line, right, in clinical trial, it's really going to cost you a lot of money. So if at all, it doesn't mean that we want a drug to fail, but if at all drug has to fail for whatever reasons of safety or efficacy or poor PK properties, or because of poor PK in human or lack of efficacy in human, want that drug to fail as early as possible, so that can save a lot of cost in unnecessarily taking that drug all the way to clinical development and then realizing that drug cannot go further, you see. So at different point, different field comes into the play. So when you decide the target, as I already mentioned, the knowledge on proteomics and genomics comes into the play. So that's the role of a biotechnology, correct, or a pharmacology student. If your understanding on proteomics and genomics is there, you can really contribute in target identification and validation. When you're doing a lead identification, your chemistry knowledge comes into the play, especially synthetic and combinatorial chemistry. So medicinal chemistry is very important. Of course, that hit binding is happening to the molecular target, so biotechnology helps. And if you have an understanding on natural products, that also helps in here, correct? Even when you scale up the synthesis for lead optimization to go into the preclinical testing from mg to grams to kg, your knowledge of chemical synthesis and formulation development, analytical chemist or formulation scientist or chemistry, synthetic chemist, all that role comes into the play during preclinical development. When you're doing a safety studies, then your knowledge of pharmacology especially helps. Even your veterinary or zoology background really helps here, and pharmacology also helps in here. And if you have studied clinical research, then your role comes into the play in designing or execution or sampling, wherever in clinical trials your uh, qualification or knowledge comes into the play, correct? So clinical product development role comes into the play. Whenever it is a manufacturing, at this stage of preclinical and even after going to phase 3, you want to manufacturing, so that your chemistry, your CMC knowledge field comes into the play. So that's why we always say, and all of you would have by now understood very clearly, that different disciplines have a role to play in drug discovery and development, be it pharmacology, microbiology, biotechnology, chemistry, veterinary, zoology, or any other related field, or clinical research. All of them have a role together to play in drug discovery and development. Yeah. So good. So I think still we have time. So we will try to just talk about in few slides about GLP, and then I will stop, and maybe we will cover the entire GLP in subsequent classes. Okay. So good laboratory practice is a quality system concerned with organizational process. Okay, and the conditions under which a study is planned, performed, monitored, recorded, archived, and reported. That's it. So you see, within this GLP practice, basically it's a quality system. So what does it mean? If I say my lab is GLP certified, means I have all the required quality systems in place in my organization or in my institute or in an academic institute, where all the processes are well defined. which are this process? As a scientist, as a student, as a researcher, you can easily correlate that. You see, whenever you want to do any study, be it a PK study, be it efficacy study, be it toxicology studies, what exactly we do? We plan the experiment, right? So what does it mean? Means, suppose if it's an in vitro assay, you would like to plan that, okay, this is the dosage I will be using, this is the concentration I'll be using, this particular cell line I want to use, this particular non-cell based models I want to use, this is the plates I'll be using, these are the instruments I'll be using. So you're basically planning. So you have to document that. So that's called study plan, right? So you have to plan your study, okay? Then you have to perform. So you should have a protocol in place where everything is defined, that take this many microliter of compound A, add to that this much of microliter of NADPH, this much of microsomes, incubate at this temperature for this much time. So that is called protocol. So based on that predefined protocol, you're going to perform. Once you are doing that, it has to be monitored, of course, because you're using some instruments, or you're using some even pipettes, automated pipettes. So you have to monitor. So we want to monitor that it's going correctly. Then finally it has to be recorded, right? So you use some instrument for, say, quantification. Then say it is a spectrophotometer. So where you have actually recorded the readings. If you're doing an HPLC, you will have the graphs, right? You will have the peaks. If you're doing a mass spec, you will have the data in mass spec, right? So based on how the experiment is performed, monitored, whatever data comes, it has to be recorded. So it starts with a study plan, protocol to do the study, quality assurance system to monitor. Finally it gets recorded, and then you prepare a report out of it, right? So it has to be archived. What has to be archived? Both the things. We'll cover this in detail, don't worry. I'm just spending some time to explain you what all things are included in GLP. So archived means sometimes even the samples has to be archived. It could be a plasma, serum, or blood samples or tissues. It has to be archived along with the study data, which includes the raw data, the study plan, study report. Everything has to be archived. So this is when you do in such an organized fashion any experiment, means you have required quality systems in place, and hence your lab can be a GLP certified lab going forward if you apply for GLP certification, right? So that's why we say the study should be done in compliance with GLP to ensure the quality and reliability. Got it? So what are the major components? I already talked about it. So you should have an SOP, standard operating procedures, on everything, on management, on the facility as such, on studies, on different techniques, on instruments. You should have SOPs for each of those procedures. There are no unknowns in GLP, in a GLP world. We want everything to be well defined. So that is called SOPs. There should be formats, right? If you want to put a data into a raw data, so there should be a format, say entry, exit, instrument logbook, data entry logbooks, data entry forms. If you deviate from the protocol, there should be a deviation form. If you want to modify the protocol while the study is going on or before the study, there should be amendment protocol. So all these things, formats should be in place. If you're using an instrument, we want that instrument should be perfectly in a working condition. So how do you ensure that? You should have calibration and validation records of the data. If you're using a software, you should have the validation data. If you have an Excel calculations to be done, even those Excel should be validated. So we ensure that the whatever instrumentation is to be used, it is calibrated, it is validated. In fact, for all the instrument, you should have a master calibration schedule in place. You should have a master maintenance schedule in place. As I mentioned, a study plan should be there, which defines all these things, that how exactly study is going to be done, be it in vitro or in vivo. Finally, a report has to be there and a document control. And then at the end of it, there should be a provision for archival. Of course, the hard copy, the forms, all the documentation. If at all you have soft copy, all the soft copy, wet tissues, slides. If you've done histopathology, if you have stored the organs, you can put the wet tissues also in archive. So all these things are very important components of GLP. So tomorrow we'll try and tomorrow or day after, let me just see. We'll try and see detailed what do you call aspects of this GLP. Okay, so that you can really understand how exactly GLP works. This is just a pictorial presentation, representation of a GLP facility, how an organizational structure are generally there. This is just for your reference purpose. The head of the facility, if it is a GLP facility, we call him as test facility management. So the head of the institute or that particular institute would be called as TFM. So in your articles or whenever you look for any research, this guidelines and all, it is mentioned TFM. So don't worry, TFM is nothing but test facility management. You should have a quality system in place. So you should have a well-defined quality assurance. The guy who's responsible for conducting the entire study, starting from preparing the study plan to reporting and archiving, would be called as study director. And you should have a separate archivist whose job is to archive all the things related to a study, correct? And study director, as you can imagine, his role is of paramount importance because he is the guy who is responsible for implementation of the study or for execution of the study. So under him, he can have a team of pathologists who will do the pathology. Okay, support facilities, all the support facilities are included under him. Actual study persons who will really conduct the experiments. Support facility, it includes, suppose you are preparing a formulation, so somebody to prepare the formulation, somebody who will do the analysis of that formulation. Suppose I say I prepare a formula, or formulation scientist says that he has prepared a formulation of 10 mg per ml. Who will prove that it is 10 mg per ml? So you need a analytical support. So guy who will take the sample and he will analyze the sample, do the quantification using HPLC, UV or mass spec, and he will say that yes, it is 10 mg per ml. So then it is ready to go. So that guy also will have to work under study director, and it comes under support facilities. If your toxicity study is having a separate satellite group of toxicokinetics where you want to do toxicokinetic studies. So toxicokinetic is nothing but when you do a pharmacokinetic study at the same dose as toxicology, it is called toxicokinetic. So low, mid, high, I have used 3 doses in a toxicology study. Same 3 doses, either single or repeated dose, I can do a toxicity study. I mean toxicokinetic study. So that is nothing but I will administer the drug at those 3 levels, and I will collect the blood samples at different time points, and I'll try to plot a graph of time versus concentration, and then find out all the parameters. That becomes a toxicokinetic studies. The people, the scientists who are going to help you, with toxicokinetic data, they are analytical scientists, bioanalytical scientists who will actually analyze these biological samples, and they will actually give you the data that this is the concentration on each of the time points. So those are bioanalytical scientists, and then finally you have the data from the study. Now you want that to check whether they are statistically significant or not. So that is done by a biostatistician. So that also comes under support facility, right? So take home messages: the institutional head in a GLP facility is test facility management, archivist, study director, and quality assurance. All of them comes very well under test facility management. And study director is the guy who is responsible for execution of the study with the help of a study pathologist. This is in perspective of a toxicology study, right? So study pathologist and all the support facilities, including formulation scientist and bioanalytical scientist, veterinary or other pharmacology people who are doing toxicokinetic animal studies, and a biostatistician. All of them as a support facilities comes under study director, and actual study personals who will really perform the experiment. So that is the setup of a GLP lab. So Okay, so I think I will stop here, and flow of GLP process I will cover in tomorrow's this thing, where how exactly the process works. Basically, don't get confused. It is pretty straightforward as it happens in any normal lab, right? If I have to do an animal experimentation, I'll be doing the same process. Only thing is, under GLP, all these things should be, all the components which I have explained should be a part of it, and all things should be documented. Okay, so it is very easy, of course, to follow GLP, but your intent should be that. I mean, if your lab is GLP certified, in nutshell, it really avoids any manipulation. Okay, so everything is really documented. So the flow of typical GLP process in a lab, I will just discuss in detail in tomorrow's lecture, or whenever we in future we talk of GLP, we'll start from this slide, because this will really define what are the steps involved. How do you really do a study plan? It goes for an approval. How you get an animals, and then how do you prepare a formulation? How do you dose the animal? How do you observe? How do you do the sample processing? How do you sacrifice? How do you prepare a report? How a quality assurance people role comes into the play. So we'll talk about it. Don't worry, and we'll cover this in detail in the subsequent class. Okay. With that, since we are on toxicology, and we have talked about much about this beautiful animals, which play a very important role. Let me end on this slide, and now you can see the person who is holding the animal. Very animal looks much relaxed, and the person is wearing gloves, which is must. Okay, and I don't know if you're able to see, but they have very sharp incisors, so you should be very careful in handling these animals. Even their nails, you can see, they're very sharp. So that's why we say that before handling the animals, make sure that you have taken tetanus as a preventive measure, tetanus injection and hepatitis B injection, and always wear uh, protective clothings and protective wear, including gloves, mask, face mask, goggles, apron, head mask, shoe cover, so that the chances of any zoonotic diseases uh, to and fro, from you to animal and from animal to you, would be bare minimum. So with that, let me end my lecture here, and would be more than happy if there are any questions. Thank you.
Thank you, sir. Students, anyone having any queries, please respond in the chat box.
Meanwhile, while they decide on the questions, let me answer one of the question asked yesterday, and about uremia. I will discuss tomorrow. But one of the question which was asked on neonates, that was a good question. That what happens about the ADME? So, as your question suggests, neonates means they are too young, right? So, of course, they do have organs well defined, but they are not fully developed. Okay, so what happens is, generally, if you consider from a DMPK perspective, the clearance is the factor which would be impacted. So generally, it's going to be low clearing compounds. Okay, I mean, much like a uremia case, but I'll talk about uremia kidney disease tomorrow. But here also the clearance may be less for a couple of reasons. Sometimes even the liver is not able to do the complete metabolism. So there could be accumulation of drug in neonates, right? So it is going to be impacted. But the question was, how about the clinical trials? So you see, you'll be surprised. Neonates are very important for because they do need a lot of treatment. Many a times there are sick kids, I mean sick infants who are born suffering from many dreadful diseases. They need to be treated. So how exactly? And you'll be surprised to know that up till recently, as recent as 2009, all this started back in 1997, but only by 2009 FDA has come up with their new act, which really defines the studies to be done in infants or neonates, right? Because that dose has to be too low. Please remember, what happens is in a newly born neonate, the blood volume is as low as in mLs, say 45 ml or 50 ml. So the blood which you can withdraw would be hardly less than 1 ml. So you can see there are practical challenges in doing so. So there are a couple of ways people have done it, and of course it is done. There is an act in place. So something called as scavenged plasma samples are taken if at all you have to do the study. So scavenged plasma is basically, as word come from scavengers, which is leftover. Like we say that pigs and all, they feed on they are called scavengers. On leftover they survive. So scavenged plasma means whenever for any diagnostic purpose or any other testing blood is withdrawn from the infants, the leftover plasma or the blood is taken and utilized as sample after a PK study or your clinical trial sample, it will be considered. So that many less samples you have to take from your actual trial. So you are trying to minimize the samples. That is number one. Secondly, as I mentioned, to do a full-fledged study is never a possibility, right? So there are different ways people have come. One of them is called Monte Carlo simulation model, which is highly acceptable model. Okay, so far it has been very successful. Monte Carlo simulation model, which works on principles of population pharmacokinetics, and that model is widely accepted to determine the ADME properties of a drug in neonates. So many a times it is not the actual study, but the data which you have from elder kids or adults, whatever you have available, that data can be utilized to extrapolate in neonates. So that is also being widely accepted. So please remember, it's called Monte Carlo simulation models based on population pharmacokinetics. And also it's equally important that whenever you do this studies, there are plenty of examples. Just one I will like to quote. One, dexamethasone, as you know, is a life-saving drug, and it was used in neonates at certain doses, and eventually it was meant for a good purpose. Like it was helping kid to prevent lung damage from happening in neonates over a period of time. Okay, so the purpose was good, and actually it prevented lung damage on a long run, or lung infection on a long run in neonates. But because of the doses they were used at, eventually it lead to neurobehavioral side effects or adverse effects in those kids. When those neonates, over a period of years, it was found out actually, when those neonates were grown up, there were some adverse effects which were noticed. So you see, it takes years together to find out those adverse effects, which was really not called for. So it was heavily criticized, usage of dexamethasone, and it's a real case study. In US it happened, and since then people are actually not using dexamethasone, or if at all it is used at a much lower dose. So in general, the principle for neonates is whatever dosage are being used, they are always used at a lower dose than the dose where it can have a minimal risk generation in human. In other words, any drug which has to be used in neonates, its entire safety study should be complete. For adults also same is the case, but for neonates, what is asked for is even additional data. So when you are developing your drug, you should have sufficient data to establish the safety of your drug, either in preclinical or even in clinical scenario. For adults also it doesn't matter, but based on that the same data can be extrapolated. So like reproductive toxicology studies must when you do a when you want to take a permission for neonate. So reproductive toxicity would be done so that those toxicity parameters have been studied from mother to the kids and I mean to the infants in embryo development. So all those studies are done. So basically, before you get a permission to use neonates for a clinical trial, there are very stringent requirement from a regulatory point of view. Only then they give you permission to use neonates. And of course, there are other challenges like nobody really allows you to use neonates, right? But at the same time, there are so many disease which happens in neonates, so it needs to be treated. So of course, there are so many efforts are going on, and guidelines are also in place. But as I'm saying, more and more safety studies needs to be generated to be done early in preclinical as well as in clinical trial before we really ask for permission for pediatric usage, especially in neonates. So you'll be surprised, but there's a term while going through, I came to know it was called as pediatric orphans. Means pediatric applications were treated as an orphan for quite some time. It's only in 2009 or 7, even the regulatory agencies have come up with some specific guidelines related to pediatric pediatric clinical trials or usage of drug in that. So hope I've been able to answer your question. And same way, tomorrow we'll take your question on uremia. So uremia again is nothing but it talks of chronic kidney failure where you find such condition. So whenever there is a renal damage, we'll talk about the impact on clearance in tomorrow's lecture. Okay. So with that, I think somebody has written GLP, good laboratory practice. Yes, GLP is good laboratory practice. So we will be covering the in detail about good laboratory practices tomorrow or day after. Okay.
Yeah, thank you, sir. Thank you so much. Students, anyone having any doubts, please put in a chat box. Sir will clear on tomorrow.
Sure.
Thank you, sir.
Thank you. Then, if there are no further questions, then would like to thank all of you, and looking forward to connect with all of you again tomorrow at 11 o'clock. So till that time, have a great day to all of you, and we'll connect with you all tomorrow. Thank you. Bye bye.
Yeah,
Thank you.
thank you, sir. Bye bye. Students, I have given the feedback form in YouTube, Facebook also. Please submit that. Based on that only we will provide the certificates. Thank you everyone.